The second part of this lecture is the question-answer session about the FightAIDS at Home project on World Community Grid. Dr. Alex Perriman will be answering your questions. The first question is, how many mutations of the HIV virus have been discovered so far?
That's kind of hard for me to ballpark off the top of my head, but there are at least 10 different positions out of the 99 positions per half. So HIV protease is a dimer composed of two identical halves. So of those 99 positions, there's at least 10 that are associated with drug resistance, and many of those positions can mutate to multiple different types of amino acids. So then you have the combinations of those different positions as well as other accessory positions. So there's a huge variety of different types of mutants of HIV protease. So in FightAIDS at Home, we focus on some of the most clinically significant combinations of mutants, especially those for which there's high resolution, atomic scale, three-dimensional data publicly available.
Are any of the drug candidates you found in the process of becoming an actual medicine?
In the process, but it's a long process and we're really just starting. So these types of fragments that I just discovered, people are doing more tests on, and some of the early allosteric fragments that we crystallized. So that was from experimental studies as opposed to computational studies, but it's been feeding into the FightAIDS at Home project. So those allosteric fragments, multiple different labs at Scripps and hopefully throughout the world are trying to develop those compounds into much more potent things. So there are many different ways that we're trying to advance these compounds and make them more potent, but nothing has yet been developed that's close enough to being an actual drug from the FightAIDS at Home calculations yet. But there's a huge volume of data that's been generated, and I've really only been able to scratch the surface and analyze just a small fraction of that. So I'll be analyzing this data for many years, and hopefully other labs will start analyzing it too. And there should be many, many, many more promising compounds that we've already obtained, but we just need to figure out which ones we need to order and test.
Another question we have is, if someone creates an AIDS vaccine, will your project still be needed?
Definitely. So vaccines are meant to prevent a person from getting an infection. So even when they eventually figure out vaccines or other forms of prevention like antiviral microbicides, which are also showing some promising developments. So even if they get prevention to work perfectly all the time against all the different strains that exist now or that ever could exist, we still need to come up with new drugs to treat the over 30 million people who already have an HIV infection. So vaccines only prevent you from getting it. There are already 30 million people who have an HIV infection, and in those patients, these new superbugs keep evolving and spreading. So even when prevention is totally figured out, we'll still need to come up with new drugs to help treat the over 30 million people who currently have it. Plus, once they have a vaccine discovered, it'll take years to test and many more years before it's systematically deployed in all the different regions that need it. So for a long time, there'll be a huge need for research in both prevention and treatment efforts.
Sort of a related question is, how many years do you think it would take to get an HIV vaccine into use?
That's really difficult for me to predict. I really focus on treatment efforts, not on vaccines at all. So I'm not up to date with all the current research on vaccines. There's definitely been some recent promising developments, but yeah, I don't really want to put a ballpark and give false hope or delude people. Somewhere in the order of years, decades, something. I'm not sure. It's a very tricky system to fight because it copies itself in that sloppy manner, so it keeps evolving new mutants. I do have hope that there'll eventually be good ways to prevent it, but I don't know how long that will take.
How many different HIV inhibitors are already in use, and why is it taking so many years to find a way to treat HIV?
Yeah, I'm not entirely sure because I only focus on a few of the particular targets, but I think there are around 30 different drugs that have been approved by the FDA to treat HIV and AIDS. I think there's around 10 against HIV protease itself. It has taken many years, but the science of the understanding of HIV and viruses in general and how human cells work has been growing exponentially over the last few decades. So back in the 80s, when you got an HIV infection, it was pretty much an immediate death sentence. Now that they keep coming up with new drugs that fight different binding sites on different targets, and they have these combinations or cocktails of different drugs, now patients are able to live many years or even decades with a decent quality of life. So there's still a lot of work that needs to be done, but there has been a lot of progress over the last few decades. A lot of people are on these retroviral treatments, and that's actually been helping to decrease the number of new infections each year in other parts of the world. But from Obama's speech today, President Obama's speech today, we learned that the number of infections in the U.S. is actually not going down. It's actually going up in some communities. So there needs to be a lot more effort devoted to it and a lot more attention paid to it, so people realize and are careful and try to prevent it, and also help donate and get the drugs out there to the people who have the infection.
Could you go over again why HIV is so difficult to treat, and do we have to keep finding drug after drug after drug forever?
Hopefully not forever. So that's one of the main overarching goals of Professor Olson's lab is we're trying to not only discover drugs that work against the mutants, or compounds that work against the mutants that can be developed into drugs, but we're specifically trying to develop these compounds in ways that make it harder to evolve drug resistance against them. So there are a few different strategies that have been shown to work in model systems of HIV, as well as some cancer cases and other diseases, where there are some strategies we can use to try to design compounds that are much harder to evolve resistance against. Also trying to find compounds that bind to new sites that are also harder to evolve resistance against. So we don't think that we'll always have to keep finding new things. That's what the Olson lab really focuses on, is designing new things that work in the face of evolving drug resistance. But it's still going to take many, many more years and research against many different types of targets on HIV before there's a good enough cocktail that can really corner HIV in sequence space to make it harder to evolve resistance and still be able to infect patients.
So is your project's goal is to treat the disease, not create a vaccine, so to keep the virus in check at an undetectable level or something like that?
Right. But also some recent data has shown that if you're treating HIV properly, that also helps prevent the spread of the infection to other patients. And that's because when you're on a good HIV cocktail that's able to suppress your viral load, it's able to stop the virus from replicating and spreading to other cells in that patient. So when you decrease the viral load, that actually lowers the probability of that infected person spreading HIV to another person. It doesn't eliminate the probability. It just decreases that risk of spreading the HIV infection. So by having new drugs that work against these superbugs, that can actually help slow down the spread of those superbugs throughout the world.
Is there any chance of finding an inhibitor that could cover all the drug targets that have been found before?
Yeah, the idea of having one drug hit multiple different targets is fairly new. It's called this polypharmacology, where you have one compound that can bind to multiple different types of targets. I'm not aware of any published research on compounds from HIV that hit multiple different HIV targets, although there's a chance that some of those could be developed against certain types of targets. It's unlikely that a protease drug would work against integrase, or the two targets we focus on now, but in the future we do plan on working on other key drug targets from HIV in collaboration with a bunch of other labs throughout the country as part of this new HIV center that the Olson lab is trying to create. So it's unlikely we'll have one drug that works against all targets, but we'll have many different drugs that can be used in a combination, in a combination therapy or a cocktail, that will be able to significantly block multiple different types of proteins or different drug targets from HIV to keep the infection low.
Have you done any research on the long-term effects of these HIV inhibitor drugs?
No, that's really something that would be more for the clinical experts and epidemiologists and whatnot. I really focus on fighting an atomic scale war against particular proteins from HIV. Those other parts are studied by other types of doctors in other labs. I can read the data, and I sometimes get some of that information from Dr. David Looney, who's part of this program project grant that Art Olson leads. But that's not my particular area of expertise.
Somebody was asking whether one of these treatments destroys your white blood cells, and are they in danger? Or is it just kill the HIV only?
Yeah, I'm not aware of that specific side effect. It's I suppose possible. Basically, on the pill bottles for any particular medication, it would have the potential side effects listed, or one could look in the PDR or physician's desk reference to see. I think most of the types of drugs that accidentally kill white blood cells are some of the cancer medications that happen to kill all dividing cells, and so they'll kill some of the blood cells as well as tumor cells, but it kills off the tumor cells more and quicker. For HIV, it itself infects a type of white blood cell, the T cell. So that does lower your T cell count, is because of the HIV infection itself, as opposed to the drugs that are used to treat it, as far as I'm aware.
Somebody's asking if the HIV integrase is still that interesting of a target. What's next?
I would say yes. I mean, that clinical data showed that when you added raltegravir to that cocktail, then that helped treat AIDS patients who weren't responding well to the previous types of drugs that didn't target HIV integrase. There are also a couple other integrase inhibitors that are in clinical trials and under development that target that active site region. The problem is HIV copies itself in a sloppy manner. If it happens to change in a way that blocks a drug, then that gives that strain an advantage and it spreads. So there are already drug-resistant mutants in the active site of integrase, and that's one of the reasons we're focusing on that allosteric site, is to develop a new class that should work against those raltegravir-resistant mutants, and that could also be used in combinations to hopefully help suppress the evolution of new mutants in HIV integrase. But it's definitely a very promising target, and it's being given a lot of different attention in different research labs throughout the world, as well as different companies.
If we can slow down the contamination rate, do you think we could eradicate the disease in 20 or 30 years?
That's once again more for the epidemiologist, a very different type of scientist who studies that. I do have hope that it can be substantially decreased. As for whether it can be completely eradicated, I don't want to try to predict that. But I did see one question that I'd really like to answer down there at the bottom about the new project. That mosquito logo is a logo designed by Dr. Stefano Forli, a member of the FightAIDS at Home team, and that's for our new project that we just launched on World Community Grid a couple weeks ago, the Global Online Fight Against Malaria, where we're going to be doing these docking calculations against targets from the malaria parasite in order to hopefully discover and develop new compounds that can fight the drug-resistant superbugs of malaria. So HIV and malaria are two of the three deadliest infectious diseases, and now the Olson Lab is going to be able to use World Community Grid to try to fight both of them. So please join us at worldcommunitygrid.org, and you can sign up to help both the FightAIDS at Home project as well as our new global online fight against malaria.
We have time for one more question here. Are there currently any target compounds that show promise against targeting reservoirs of latent HIV virus?
I've seen a few press releases about some promising data on that. I don't think anything's close to being potent enough for the clinic yet, or for clinical trials. I don't focus on that particular type of research, but there are people who are trying to figure out how to either find the latent hidden versions of HIV in those dormant cells in the reservoirs of the immune system. The problem is, so since HIV infects T cells, you can have an infected T cell that lies dormant. So the way that the immune system works is once you're exposed to antigens, if you have the B cells and T cells, the white blood cells in the immune system that fight off infection, so if those B and T cells happen to produce the right types of antibodies or other things to be able to kill off a certain type of infection, like chickenpox or something, then those cells are kind of immortalized and go dormant, and then they hide in these reservoirs. And normally that's a good thing, because if you're then exposed to chickenpox 10 years later, you can mount a good immune response quickly to fight off that infection that your body already knows how to fight. The problem with HIV is, HIV could infect one of those anti-chickenpox T cells, and then that T cell could be dormant and hide in that reservoir until you're exposed to chickenpox again, and then that T cell would wake up and start copying itself, and then those would be infected cells. So the whole idea of reservoirs and immunologic memory does make it very, very difficult to permanently cure an infection, because the HIV can just be hiding in our DNA, but not really producing the HIV proteins that the drugs work against. So it's a very complicated issue.
Well, thanks again, Dr. Alex Perriman, and those attending. Sorry if we didn't get to your question. Please use our forums on worldcommunitygrid.org to post any additional questions. This concludes the webcast.